Well, thank you so much. It's really a great pleasure to be here. I'm very excited to learn about a lot of the things that you do. I come from the world of basically a lot of mice, but we all have a role to play, I always believe, and we really look forward, of course, to clinical translation. So I'll just turn this here. Okay. So as you all know, of course, diabetes really is a disease that for many people, whether they have type 1 or type 2, they really can have this disease for many, many years. And diabetes is a chronic disease. And of course, as we know, there's a heavy emphasis on how to treat the hyperglycemia. But the real problem with diabetes is the complications that the people get. And it's not so simple to say, well, just keep the hemoglobin A1C perfectly normal, because as I've learned with my clinical colleagues, there are some patients for whom this doesn't work because their glucose levels go too low. And when they go too low, that's almost just as bad as really having hyperglycemia. So the work that our lab has done then was to try to ask the question, what are the mechanisms by which hyperglycemia exerts its maladaptive effects? And if one can understand what those cadre of mechanisms are, that could lead then to the identification of therapeutic opportunities. And so in the world of diabetic complications, we ask, well, is high glucose so bad? And as you know, whether you have type 1 diabetes or type 2 diabetes, there really is a plethora of epidemiological research that basically says, yes, it is bad, because glucose affects not only the large blood vessels, those being what we call macrovascular complications that lead to heart attacks and strokes, but also it's the microvascular complications that lead to morbidity and mortality, and very, very importantly, a loss of quality of life. Renal failure is not a very good way to have to live with respect to frequent dialysis. Blindness, of course, is a very terrible complication. And it turns out my clinical colleagues will tell me that one of the main things that people with diabetes fear is becoming blind. And they fear amputations, and they fear things that will really change their sense of who they are. And so the way that our laboratory got involved in this is because when we first started looking at diabetes and complications, we were a biochemistry lab. And as you just heard from Tim, we were learning about advanced glycation end products, and that these products clearly increase in hyperglycemia. High glucose drives this non-enzymatic glycation reaction, and it is well established that people with diabetes have a significant increase in heart attacks and strokes, not only in the numbers of these events, but very importantly in the severity. A diabetic person has been shown in many pathological studies, unfortunately, to have larger infarcts, and therefore there's something inherently about having high glucose that leads to this. And why is this the glucose? Well, one of the main hypotheses is that this predilection to complications is true whether you have type 1 diabetes, which of course is a relative deficiency of insulin because of attack of the immune system on the pancreatic beta cells, or type 2 diabetes. Type 2, of course, often is characterized early in the course by high levels of insulin because there is insulin resistance at the level of the target organs. And so this slide we had found many years ago, and it's very, very appropriate because what it basically says, and what I say when I speak to people that have diabetes, it's really a head-to-toe disorder. So no matter where you're thinking, you have blood vessels. And when these blood vessels undergo dysfunction, which is one of the earliest signs of the development of complications, head to toe, it really affects people that have diabetes. And so it's very interesting that over the years, the biochemistry of the advanced glycation end products early on, of course, focused on the fact that they can form, first and foremost, endogenously, and that high glucose is a driver to the development of these advanced glycation end products. And Obviously, what has been shown by many biochemical studies is that the higher the levels of glucose, the higher levels of glycosylated hemoglobin, this usually correlates to having greater AGE burden. So in some ways, measuring the AGEs actually is a long-term barometer of measuring the effects biochemically of high glucose in the system. So this unfortunately complicated slide basically is very important to show because essentially what it shows really is how these ages form. And so in terms of the entire pathway, what you start off with are actually reversible products. So hemoglobin A1C is actually a reversible product. It's an Amadori Schiff product that is a glycation on hemoglobin. What advanced glycation end products are these different products that occur after a number of different series of various biochemical reactions that lead to these ages. And it's really important to note that ages are very heterogeneous. They can form on all different types of proteins and also on lipids. And the bottom line is that they have two major characteristics, either the non-fluorescent ages, and then there are the fluorescent ages. And fluorescent ages probably are derived from the fact that one of the biggest consequences of these ages is their ability to post-translationally modify proteins of the basement membrane. So collagens, these types of proteins, when they can become glycated, often they take on fluorescence. And so it's that characteristic of fluorescence that if you've heard of measuring skin autofluorescence, that is the basis of this. And in certain places throughout the world, they do this to be able to try to understand the long-term effects of these glycation products. Because when you're looking at skin, obviously you're looking at a lot of collagens, a lot of basement membrane type proteins, and therefore this entire idea is that skin autofluorescence may be an indirect measure of someone's AGE burden. And of course, there are many papers that show that people that have diabetes have more of these. So the bottom line, it's real. Back when the type 1 diabetes community was working on the DCCT edict study, which was a long-term follow-up study that asked a very simple question many, many years ago. It's very interesting to come up with this important question so far before they even had all of the advanced forms of insulin that we have now. These were type 1s. And basically the bottom line question was rigorous control of hyperglycemia better many years later than just random, just checking your finger stick every so often, etc. And it turned out, of course, that the answer was the former. It was better. And then over the years, they started adding surrogate type studies to DCCT edict. And one of them actually was a skin biopsy. So of course, there was a lot of worry, well, can we be doing biopsies on skin in diabetic people because of wounding? And the bottom line, it was a punch biopsy that they took in the underarm area. And many researchers, such as Vincent Monnier, were able to show that there were significant accumulation of ages in that diabetic skin, and that age accumulation was higher in the people that were in the general treatment group, not the strict glycemia. So that's a long-term measure that basically says ages accumulate, and they very likely are due to the long-term effects of hyperglycemia. And so what was quite interesting was over the years, and Helen Vlasara, who previously worked at Mount Sinai, was one of the people who actually looked at this very, very closely, the ages that come from foods. And Helen had developed an entire assay, mass spec-based assay, where they looked at all different types of foods that humans eat. And basically what she was able to show was she characterized foods based on high AGE content versus low AGE content. And so I went through the literature and I found multiple examples of what were typical high age foods. And of course, you may all know that the prototypic photo is that guy at the barbecue with all the steaks and all this. And then, of course, this thing called creme brulee, which has those are ages, So, of course, they're delicious. But on the other hand, that's pure AGEs. So the bottom line is that there are certain foods that are known to have high age content, and then others on the other side, I see everyone walking around with the legume shirt, and in fact, these things are very low in ages, and obviously you can tell that there's a bit of a difference with respect to, how tasty some of these things might be, depending upon your viewpoint. But the bottom line is that ages can also be imbibed through foods. And a number of laboratories, not just Blasara's, actually showed when they fed mice high-age diets that all of the different complications, these animals didn't have diabetes, but the complications of the ages looked a lot like what happened in the diabetic animals. And the reason for that was that at least in part, it's very likely that hyperglycemia, endogenous-driven ages, are really important in the pathogenesis of complications. And so what was very nice, in going through the literature and watching all of this over the years, is that a number of laboratories said, well, in human subjects, so, it's great to be studying these mice, but can we change things, as you just heard, in human subjects? And so essentially what a lot of these different studies did, this is one very, very nice one 2 years back in Scientific Reports, and basically what they did in the humans is they were randomized to low-age diet and high-age diet, and then they looked at different factors in these people that were known to be risk factors for all sorts of cardiovascular and other complications. And what was really interesting is that even though there were no changes in body weight per se on a high versus low age diet, because they matched them in order to take away that variable, what they found was that there were benefits in terms of cholesterol, metabolism, and very importantly, here they used the HOMA-IR. They didn't clamp these people, but with the HOMA-IR, they were able to show that their insulin resistance actually improved. And I thought that this was very interesting because you just heard the word there about inflammation. So of course, what they looked at in these people was what happens to some of the inflammatory factors. And obviously, you can take a lot of sources. You can look at blood, plasma, serum, peripheral blood mononuclear cells. And basically what they saw was that markers of inflammation, and a very prototypic one, tumor necrosis factor alpha, decreased on the low-age diet. The isoprostane and ages, these are examples of oxidative stress. And also, the soluble RAGE, which I'll tell you more about, was also modified in these low-age diets. They also found that adiponectin actually rose, and of course, adiponectin is a molecule that's involved in good adipocyte function. So, the question then that our lab asked many years ago was, well, how do these ages exert their effects? So the very simple hypothesis was that these ages might just be biomarkers of high glucose. But it was also intriguing to say that if they can post-translationally modify basement membranes and collagens and this sort of thing, did they actually have the ability to interface with the vasculature? And the question was, did they interact with cell surface receptors? In other words, a gain of function. So even though we have normally albumin circulating in us and immunoglobulins, if they're glycated and aged, is that a gain of function? And one of those gains of function might be the ability to start to be recognized by cell surface receptors. And so, toward that end, we actually did very simple experiments. We knew that if we took albumin and we put it in high levels of glucose, or we put it in normal levels of glucose and put it into an incubator at 37 degrees, just these proteins, we would see that after a week, 2 weeks, they actually started to brown. And the browning was the fluorescence because they were generating the ages. Whereas in the low glucose control, they really never became brown. It was very interesting, just to look at that question ex vivo, so to speak, and we saw very clearly that they were glycated. And what we did then is we radio-labeled these age products and also the control. Obviously, the controls are usually more important than the test question. And we found that only the glycated protein material could bind in a dose-dependent and saturable manner to cultured endothelial cells. Back in those days, we used to wait on a little line in front of the delivery room, and we would get the cords, and then we would isolate the human umbilical vein endothelial cells from the cords. And so these were great because they were human, and obviously because of the methodology, they could be pure, so they weren't a mixture of all sorts of things. But the most important point was that you could destroy that binding by treating those endothelial cells with proteases, for example. So what did the proteases do? They destroyed vulnerable proteins based on the different sites where these proteases could act in the protein. Once we did that, the binding was abolished to the age products. And then it took a few years, but essentially what we did is we just kept narrowing down in this endothelial cell mixture using all kinds of biochemistry approaches to figure out where was that AGE binding property, where did it come from in that mixture, and then ultimately, you know, the very hard to do, but ultimately we were successful, we were fortunate, was a single band on a gel that was presumably the quote-unquote receptor for these ages, and then it went off for sequencing, and it turned out that it was a newly described member of a very interesting family of molecules, and that family of molecules was the immunoglobulin superfamily. So the immunoglobulin superfamily are molecules that are involved in innate and adaptive immunity. And so what is this molecule doing in the mix of this? And I have to say, even many years later, we still don't exactly know the answer, but I think with the research tools that have been developed, we do have a chance in the next 10, 20 years to get to that answer more closely. And so rather unimaginatively, I must admit, we call this receptor for advanced glycation end products, or RAGE, and then that name basically stuck. You know, maybe it wasn't the best, but what we did find over the years, and the reason why this name is a little problematic, is because not unlike other Ig superfamily molecules, RAGE could bind to other ligands. And essentially, we used a very similar approach, a biochemical approach, to find out the opposite question. What does this RAGE bind to? And what was so interesting is that when all these different hits came out of the sequencing, they were all molecules involved in inflammation. For example, S100 calgranulins, high mobility group box1, amyloid beta peptide. And this was actually, you know, what does this have to do with diabetes? And in fact, a lot of research over many, many laboratories over many years really suggests that even diabetes is essentially a pro-inflammatory state. And we knew that this was an Ig superfamily molecule because the molecular cloning indicated that the extracellular domains were composed of immunoglobulin-like molecules. And in the laboratory, while we were working towards identifying potential therapeutics, we also use the extracellular domains of RAGE, which we call soluble RAGE, because we didn't have this hydrophobic piece here. And these soluble RAGEs actually ex vivo, when they were administered to mice, were able to bind up the RAGE ligands. So it was an oversimplified idea of how to make a therapeutic, but it turns out that in the beginning years, it was a tool to be able to ask the question, the very important question, so what? So this thing binds to the endothelial cell on the ages. What does this have to do with anything? And obviously, that's how you determine if it can be a therapeutic. And so this slide actually was a very interesting. I was looking around to find, you know, things that would also help us to understand not only the endogenous ages, but here you also see that this concept with respect to the RAGE by these investigators, they also use the idea that the food-derived ages, when they enter into the circulation, or whether they're formed endogenously, they can bind to this RAGE molecule. And many different laboratories now over the years have shown that RAGE basically triggers an inflammatory response and cellular dysfunction. And at least in part, in all of the different cell types that are vulnerable to diabetic complications, this is what we see. And so obviously this soluble RAGE turned out to be a very important tool. So many years ago, of course, when this thing was first identified, we didn't have antibodies, so we had to generate them. And then as the first papers came out, it was wonderful. People in the translational science started to ask, well, I have some diabetic tissues with this or that, and I would love to be able to try those antibodies and see whether there's more of this RAGE in diabetic tissue versus non-diabetic tissue. And Francesco Cipollone was one of the first people to look at human carotid endarterectomy samples. And what Dr. Cipollone found was that diabetic people had higher expression of RAGE in their atherosclerotic plaques compared to the non-diabetic people. He also did Western blots to basically be able to be sure about the size of the protein and what the staining was actually showing. But essentially, it went along with what we were seeing in the mice. Very low levels of expression of RAGE in homeostasis, but then in diabetes, upregulation of RAGE in multiple tissues that are targeted for dysfunction. So the lab started out asking about accelerated atherosclerosis, and indeed, these were the first experiments. And in the years when this was first identified, parallel work going on in other investigators' lab was basically developing mouse models of atherosclerosis. And that was important because mice have very high HDL levels, and they basically don't get atherosclerosis. Even if you feed them horrendously cholesterol-rich diets, they don't really get atherosclerosis. And so, two different investigators generated mice that were devoid of apolipoprotein E, and these animals spontaneously develop atherosclerosis. And Nobuyo Maeda shared those mice with us, and she understood that what we wanted to do was to make them diabetic, which we did with streptozotocin, and we found that when they were diabetic, that their atherosclerosis, easily measured in their blood vessels, that's these white plaques, was very much accelerated by the hyperglycemia. And so I already told you that the extracellular domains of the RAGE are the soluble form of the receptor, and with Jean-Luc Wattier in Paris, we radio-labeled this soluble RAGE that we generated recombinantly in the lab, injected it into the mice, and then with Jean-Luc, we found out that if we gave this soluble RAGE by intraperitoneal route, that the half-time of elimination was 96 hours. And it's interesting, it was longer in the diabetic mouse versus a non-diabetic mouse, presumably because it was binding to all those ages on the cell surface. But that half-life basically allowed us then to administer it once a day to the mice in order to ask hypothesis-driven questions about whether the soluble RAGE could mitigate the effects of accelerated atherosclerosis in a diabetic mouse. And so basically what we did is we took these animals, we made them diabetic. Citrate is the control for the streptozotocin. And we found again that after 8 weeks of hyperglycemia, the atherosclerosis was advanced. This was baseline. And then in a different group of animals, of course, we then treated those animals from 14 weeks to 20 weeks with either soluble receptor or mouse albumin as a control. And over many mice, this is just a representative image, what we found was that the soluble receptor was able to greatly attenuate the advancement of the atherosclerosis in these mice. And very importantly, the soluble RAGE had no effect on the levels of glucose, and it did not affect the cholesterol levels or the triglycerides. So presumably, the way the soluble receptor was working was not by changing the quote-unquote risk factors, but by diminishing this inflammatory response that this diabetic blood vessel was demonstrating. And others were working on this as well, and they showed that hosts and hosts of oxidative stress molecules, inflammatory molecules, are enriched in diabetic atherosclerosis, even in a mouse. And so the soluble RAGE obviously has limitations in terms of how you can fully interpret such data. And at the same time we were generating soluble RAGE, we also were developing the homozygous RAGE null mice. So these are mice that have no RAGE whatsoever in them. And it was very fortunate because unlike what happens sometimes, these animals had a normal lifespan and they were able to be fertile. They lived to adulthood, and it allowed us then a model to be able to ask, well, if the atherosclerosis-prone animals don't express RAGE, what happens to the atherosclerosis? And very importantly, what we found is when we looked at the double knockouts, so the mice that have no ApoE and no RAGE, what we saw was that the atherosclerotic lesions were very greatly attenuated. And again, glucose levels, no difference. And cholesterol, triglyceride, no difference. But what was different was when we looked at the inflammatory cell content, for example, macrophages and T cells, we found that these components of inflammation and inflammatory molecules themselves were greatly attenuated in the RAGE-deficient mice. So over the years, you present all these different things, and then the scientific community started to say, well, this is very interesting, but in the peripheral arterial system, it's actually critical that you do have an inflammatory response in order to tackle ischemic injuries that can occur in people with peripheral vascular disease. And so they queried, well, what happens when you knock out the RAGE? Is there an adverse effect in this setting? And that's because peripheral arterial disease with femoral artery atherosclerosis is actually like a chronic wound in many ways. The pathology in peripheral arterial disease is apparently different than that in the cardiovascular system, in the coronary arteries. And for this reason, we developed an animal model to ask the question, well, what happens to animals that are undergoing ischemia to their hind limb? And this is done by ligating the femoral artery. And essentially what we found, this here is laser Doppler blood flow imaging, and it was very nice because what we were able to show was that after 2 months of diabetes, these animals were able to attenuate their blood flow, just like what would happen in a diabetic person, but obviously over a longer span of time. But after the ligation, what we were able to observe is that the diabetic mice devoid of the AGER, that's the gene name for the RAGE, actually had a better recovery of their blood flow compared to the wild-type animals with diabetes. And this slide basically demonstrates what is angiogenesis, or the generation of new blood vessels. And here what we saw was that the RAGE-deficient animals had a significantly greater increase in the blood vessel formation. So this result was then really important because it says we're not adversely affecting the wound healing response in angiogenesis, but we're attenuating those inflammatory factors that block effective angiogenesis. And so this slide basically is just a recapitulation of many of the work that was done, really not just by us, but by many laboratories, where we shared with them either soluble RAGE or RAGE-deficient mice to basically show that in microvascular complications, there were benefits to blocking RAGE or to deleting the RAGE entirely. And so it was quite interesting over the years, just reading the literature, to ask, these ages are really very interesting species. And it turned out that a very beautiful paper that was published in 2014 in ATVB looked at human people with obesity that did not have diabetes. And they looked particularly in the adipose tissue because their hypothesis was, well, maybe these ages can also cause insulin resistance. And so it was really important that these people that they enrolled in their study did not have diabetes. Because if you're going to be looking at hyperglycemic people's fat, then obviously there could be multiple reasons to generate those ages, like the high glucose. And instead, what Gaines and al. Showed was that these age factors were actually higher in the obese fat versus the lean fat in the people. And in parallel, they looked at RAGE levels in this adipose tissue, and they were able to show that RAGE expression was significantly higher, not only in the immune cells, but also in the vascular cells and in the infiltrating inflammatory cells. At the same time that Gaines and al. Were doing that work, we were looking at this question in mice because we were aware of this idea that ages also increase even in the absence of high glucose. And in fact, we saw that when we would feed mice a high-fat diet, which means 60% of their kilocalories from fat, normal mice eat 13% kilocalories from fat. In the metabolic organs, the age levels increase. This is liver. And also, I told you about high mobility group box 1, also increases in the high fat versus the low fat fed mice. And so what was very interesting and totally unanticipated was that when we fed wild-type mice or RAGE-deficient mice this high-fat diet, we were very surprised to find that the mice that were devoid of RAGE compared to the wild-type were basically protected from diet-induced obesity. And again, there's ages accumulating because of the high-fat diet, so the hypothesis could have been that perhaps this age-RAGE interaction contributes to the development of the obesity. Now we also looked at, we did what is called a DEXA scan, and we were able to show that the body fat, which increases greatly in wild-type animals fed the high-fat diet, was significantly lower in the RAGE-deficient animals, and this was in parallel, of course, with an overall lower level of body weight. We also looked at the glucose levels, because even though these animals don't develop very significant hyperglycemia, they do develop glucose levels in about the 150, 160 range, which is abnormal for a mouse. But the animals that were devoid of the RAGE on the high-fat diet had much lower levels of glucose. So we also looked, because one of the things, like I told you about the HOMA-IR in the human subject testing, we also looked at how sensitive these animals were to insulin. Normally, when you inject insulin into a human or into a mouse, the glucose levels will go down. But if you are insulin resistant, there will be a resistance to the lowering of the hyperglycemia. And in fact, what we saw, so the higher the bar here, that means the more insulin resistance, because the greater area under the curve. But the RAGE-deficient mice were much more insulin sensitive, even though they were eating equal amounts of the high-fat diet as were the controls. And this is really a very important question, because maybe they just don't like it, don't eat it, but in fact they were eating the food. And the mechanism that we identified was that the way that it was happening is that these RAGE-deficient mice were increasing their energy expenditure. And that's how they were able to eat this high-fat food but not develop the obesity. And so we looked at the fat tissue, because there's a lot of literature suggesting that inflammatory cells in fat contribute to the development of insulin resistance. And so even though at this stage of the game, admittedly, it's very descriptive and not a mechanism, we did see that the RAGE-deficient mice on the high-fat diet had much less immune cells in their fat tissue when compared to the wild-type animals. And so now we're working to try to understand what that means. And one of the very first questions, of course, if you're talking about energy expenditure, is the adipocyte, the fat cell itself. And so luckily, we were able, after Hurricane Sandy that thoroughly destroyed the lab, we were able to bring back a new line of mice that was something really important, because the mice I told you about so far had no RAGE anywhere. So every single cell, no RAGE. This is not really a way to understand why, just that. The way that you have to do this is to knock out this receptor in unique cell types and then say, what is the contribution of that cell type? So here, what these are animals that had deletion of RAGE only in their adipocytes. When these animals were fed the high-fat diet, so these are the ones called the CRE+, and these are the ones with the knockout. The ones with the CRE- are basically wild type, means that RAGE is expressed everywhere, including the adipocytes. And what we saw was that the animals that did not have RAGE expression in the adipocyte had significant protection from diet-induced obesity. They also had less development of the adiposity on the high-fat diet. That's the green bar. The red are the controls. And very importantly, these animals, the green bar, increased their energy expenditure. So this gave us the sense that at least part of the mechanism by which deletion of RAGE increases the energy expenditure is through the adipocyte. So the problem with this mouse tool is that this knocks out the RAGE in brown fat and in white fat. So then you can't say, well, where did this come from? What is this effect? We knew that the RAGE was expressed in the brown fat cells and also in the white fat cells. And the way we addressed this mechanistically is we did transplants in the animals. And when we did transplants, we just took normal mice called wild type and gave them either normal RAGE expressing adipocyte fat, or we gave them knockout fat, and we were really surprised to find that these animals, even though they were wild type, just bearing this adipose tissue from the deleted animals, again, the green bar, these animals were protected from the diet-induced obesity compared to the animals that received the control fat. Now, it also turned out that if we gave them the white adipose tissue, the subcutaneous adipose tissue, the effect was, as you can see here, not as good as in the brown fat, but still there was a difference with respect to protection. So what this mechanism is really exciting to us. It's a question of the next steps in the research, exactly how it's happening. So one of the problems with soluble RAGE and targeting those extracellular domains is that the extracellular domains of RAGE are heterogeneous, means that these different ligands can bind at all different parts of those extracellular domains. So how do you make a drug? If you're going to make a small molecule or an antibody, it's impossible, because if you just block one part, what happens with the others that are exposed? And so we asked the question, well, perhaps the way that we have to do this is by targeting what happens after the ligands bind. And so that means the signal transduction. And so basically what we did was we tried to identify molecules that the RAGE tail could interact with, and we reasoned that if we could block that interaction, we could stop all of the downstream negative effects. And from an experiment called a yeast-2 hybrid assay, we identified a very intriguing molecule, which is called DIF1. And so this DIF1 has lots of interesting and common properties to RAGE. One of the most interesting things to us, the two actually, is that these molecules, which are part of the family called formins, are involved in cell migration because they can affect the way actin works. They also are involved in signal transduction because they can bind through these molecules called Rho-GTPases, which basically can affect all of the downstream factors that are necessary in order to upregulate oxidative stress, upregulate inflammation. So it was a way then to say, well, if we could block this a bit more distally, so to speak, could we have a way to be able to attack diabetic complications? We were able to show, looking at this particular slide, what this shows is that the RAGE tail and the DIF1 co-localized, because when they meet, the red and the green will form a yellow. If they do not co-localize, they will stay red or green and not show that they merge. We also did experiments very critically, just to show you, I'm going to skip this. We also did experiments with my colleague Alex Sheckman, and Alex asked a very simple question. He's a structural biologist, an NMR spectroscopist, and Alex asked, well, does this DIF actually bind to the RAGE tail using these physical chemistry types of tools? And Alex, in fact, was the first to show, yes, it did, and Alex also identified the specific amino acids in the RAGE tail where the overlay of the DIF occurred. So this is his NMR spectroscopy, and essentially what he is concluding is that these two amino acids in the RAGE tail, arginine 5 and R5Q6, right, glutamine, that these two amino acids are required for the interaction of RAGE tail with the DIF1. And so what Alex and I decided to do then was to ask, well, is it possible to develop a screening assay to try to block this interaction? And so this in the pharmaceutical world apparently is a very difficult question because it means blocking a protein-protein interaction. RAGE is not an enzyme, obviously, and so therefore, is it even possible to be able to block this interaction? And so with Alex, our lab and his lab developed a assay in order to be able to, firstly, of course, we could show that in this fluorescence binding assay that when you label the RAGE tail with green fluorescent protein, that it binds to DIF1. And then, over the course of a number of months, we screened 59,000 compounds from a library to ask, well, can we find something in here that will inhibit this interaction? And so it was sort of astonishing that at the end of it, we identified 13 molecules from the 59,000 that appeared to be able to block this interaction. And then at NYU, they engaged the collaboration of a medicinal chemist who then looked at these 13 molecules And basically, we are on a long course right now to trying to be able to refine them, to develop a primary series, a backup series. Right now, we use them in the lab for proof of concept testing, but we very much hope that these molecules may someday have some benefit for people. They're far from that, obviously. We were naive a bit to understand exactly what it takes to quote unquote make a drug. But, you know, I think with perseverance and dedication, determination, anything is possible is what I always hope I can believe. And with the two different series that we had identified, the medicinal chemist calls them Series 1 and Lead Series 2, we were able to show, we did experiments that I had done like literally 20 years ago with the soluble RAGE, basically to show that if we injected normal healthy mice with our AGEs, right, by their tail vein, and then we treated them with these RAGE inhibitors, that we were able to attenuate inflammation in a range of different organs. So this work was published in 2016, and of course, well along the way, what we're working on very hard now is the refinement of these to be able to make them safe, make them accessible, make them highly potent, and certainly, you know, we do acknowledge all of the support from the NIH and other funding agencies, you know, to be able to study the basic biology of RAGE, but we do, without question, have this drive to be able to say, can we translate this? And so, just in closing, I just want to acknowledge the current members, the key alumni of the laboratory. I mentioned a number of our key collaborators, and of course, we would be nowhere without the really strong support of the funding agencies that have helped us to study this receptor over the years. So I thank you so much for the invitation and for the opportunity to present. Thank you.